We should be going. So good morning, United States. Good afternoon, Europe, and whatever other time you have in the rest of the world. My name is Carsten Schradi, and I'm a researcher in Strasbourg at the CNRS, one of the 3 co-hosts of FINE. And it's a great pleasure for me to introduce today Christian Pirk from the University of Pretoria as our presenter. Who is going to talk to us about social insects, about honeybees. As you know, one of the, in fact, most social species we have in the world. And Christian has a very impressive CV. He has more than 150 publications with more than 4,500 citations. And he is currently, and I asked him if this is true because I couldn't believe it, what's written on the homepage, he's currently supervising 9 PhD students, being the main supervisor of 5 of them. So he is supervising at one time more students than many people do during nearly their entire career, which is quite impressive. He's originally from Germany, where he did something that is called Staatsexamen, which means to be able to be a teacher, but it's equivalent to a MSc. And nearly immediately after his exam there, that was in 1999, he went to South Africa, and he got the opportunity to start a PhD on social insects. Already before, he worked on ants, and he started to work on the Cape honeybee, which is a very interesting species because, in contrast to the European honeybee, which is the same species, by the way, but a different subspecies, the workers can reproduce by laying diploid eggs. So that's quite interesting. And then he started his PhD immediately, which he finished in 2002. So I didn't ask him exactly if it was 2.5 years or 3 years, but very, very fast. So he was there working very productively. And afterwards, he did a PhD, postdoc, coming back to Germany, to Würzburg, spent some short time in China. And in 2005 came back to Pretoria, where he is based now, working on behavioral, chemical, and molecular ecology of insects, and especially social insects. Like I said, he has these 9 PhD students that are, most of them, not all of them, are funded via the African Regional Postgraduate Program on Insect Sciences, a program that the main funders come from the German Academic Exchange Program and from Sweden or some other European countries. And they have a lot of students, mainly from Africa and from all African countries, from countries in East Africa, West Africa, Southern Africa, the best of the best, working on different species in applied and basic sciences. Like a lot of them work in Kenya, mainly on mosquitoes. Another big group works on social insects, especially in South Africa. So quite a big group and very international program in Africa. That's very impressive. And when you talk to him, you quickly realize what broad knowledge he has over all these different questions from chemical ecology up to behavioral ecology. So, and now he is disappearing, but I'm pretty sure he will be back in just a moment. And yes, so I would, so Christian, I think we're all looking very much forward to your talk. And if you can share your screen now, and yeah, we'll be looking forward.
Thanks a lot for the kind words and the introduction. Yeah, it's a great pleasure to be able to showcase some of the work we are doing here in Pretoria. Or I have to say, despite the fact that I'm the one presenting the work, it is the work of a lot of people, a lot of students and a lot of co-workers. And I would like, to mention Robin Crewe and Abdullah Yusuf as 2 of the ones which actually drive together with me the Social Insect Research Group, which we have at the University of Pretoria. The title is quite long and quite, I think, descriptive, because I wanted to make sure that I'm not not using terms which are open to too much of interpretation. Because if we look at honeybees, for example, The conception was always that there's only one ruler in a honeybee colony, and until 16th century, actually, until a Spaniard actually published a book who actually stated that the leader of the honeybee colony is female, it was perceived from Aristotle's times that the leader of a colony is actually male. And the first book in English language was actually done by Charles Butler, roughly 70, 80 years later, where they actually confirmed that the queen is the leader of the monarchy. And it's nevertheless quite funny how these kind of misconceptions lead to quite interesting papers, because even after it was described that the leader of the colony is on a honeybee colony is actually female, there were especially, I can remember 1 or 2 German articles from the 17th or 1800s where they really tried to convince the reader of the importance of the males and the king in the colony and how important is for the rule. So I want to look at one of these assumptions which we have about a model organism like honeybees. For example, that there's only one female reproducing, in that particular case the queen, and all the other females are sterile and not reproducing at all. And that is actually not the full picture which we have. I tried something in this presentation, and you will see these QR codes all around. That links you, that should bring you directly, if you use it with your cell phone, to the reference. If it's one of our references from my lab or from Robin Crewe or Dr. Yusuf, it will get you to the ResearchGate profile, so you can download the PDF straight away if you want to. Otherwise, it gets you to the link to the journal itself. So if you look at the honeybee colony, the concept is that we have a queen. That's her here on the right-hand side. She is attended by the workers. That's one of them. And she is the only one reproducing, the only female reproducing. During certain periods of time, you have actually drones or males. That's this one with the bright, big, or with the big eyes. And actually, their only function in the colony is actually to get the sperm from a colony to the next virgin queen. That's their only role, and if that is done, they die, or if they're not successful after a time, they get actually kicked out of the colony. So if we look at it, we have a whole set of, at any given time, the colony is mainly consistent of females. Eggs are laid in combs, so you see the combs here. There's an egg in there. Here we have a zoom in, that is one of a honeybee egg. And fertilized eggs develop into females. Since the queen is the only one which goes into a mating flight and actually collects a sperm which she can use to fertilize, she's the only one who can lay fertilized eggs. And unfertilized eggs normally develop into males. If we look at the capacity, a queen can lay between 1,500 and some numbers are going up to 2,000 or 2,500 eggs per day. Laying workers, so if one of the other females decides actually to lay, they can lay a few eggs per day max. So what I want to emphasize already, that if we look at the honeybee society, there might be only one queen, but all the other individuals have technically all the requirements to reproduce as well, or to produce eggs. If we look at the literature, for example, even in European honey species, Fisher showed that up to 7 eggs of the eggs are actually laid by workers. That was in European subspecies. Here on the left-hand side, you see a comb with a lot of eggs laid by workers. Since they're not so nicely laid, we know that it's workers, and the queen would only lay 1 egg per cell. So there's organized. Up to 2% of the eggs which hatch, that was as well in European subspecies, is derived from workers. So there's quite a substantial number of worker reproduction going on in that society, which actually normally is defined by the fact that only one is reproducing. If we look at African honeybees, the work done by Moritz, Bayer, and Hepburn, they actually showed that you can pick up a contribution of laying workers to the population fitness, and that their contribution is therefore not insignificant, they argued. We find as well evidence throughout the literature that some colonies are not policing policing is used as a term where other workers prevent reproductive workers from reproducing by eating their eggs, but there seems to be colony or phenotypic variability as well. And we find that not only in African bees, but as well even in European subspecies in China, for example. So if we look at a honeybee, then on the right-hand side, that is from Sommerdam 1675, that is the reproductive organs of a honeybee queen. So here you have the ovaries, here you have the spermatheca, that's the organ in which the queen actually stores all the sperm she collects during their mating flight. And she can, as I said, lay several thousand or several hundred eggs per day. In comparison, you see the ovaries of a worker, drawing by Swammerdam. One of my master students looked actually into how ovarian activation is triggered to a certain degree. And what we see here on the right-hand side in these figures A, B, and C is inactive ovaries. So you see nothing at all. Intermediate, you see slight bubbles, which could develop into eggs. And then you have active ovaries where you see fully grown eggs as well. So to activate or to be able to lay eggs, you have to activate your ovaries, obviously. So you have to develop from A or to move from A to C. So how can that happen if we look into the physiology of it as well? If we look at that comb up here, you see bee bread, that is pollen collected by bees in these cells. So there's a pollen source, a protein source, which the workers could actually, like this one, just feed on. Or like the honeybee queen, she gets attended by her court, who clean her and feed her. They could actually receive as well jelly. So that's produced by a gland in the head, which the workers use to feed larvae and the queen, and potentially other workers as well. So they exchange that via prophylaxis to ensure that information flow is given there as well. So Mark Schäfer, my master student at that time, now he is head of at the Federal Institute in Germany, looked at what kind of source they're actually utilizing. So we put honeybee colony or honeybees, in that particular case, 100 of them in cages, like you see on the left-hand side. They get a piece of comb where they can rest on, and then they get food sources which they can consume. And what we see, if we look at the 3 different stages, inactive, intermediate, and activated ovaries, and compare that with the gut content, the ones which have inactive ovaries have a really huge hindgut content, suggesting that all the excess of pollen which they had to digest is actually accumulating in there, and they actually have to fly out to get rid of it. Whereas workers which activate their ovaries correspond to an empty hindgut, which suggests that they actually received the jelly from other workers. And indeed, if we look at the numbers itself, undeveloped ovaries or inactive ovaries, 60%, over 60% has large amount of pollen in the back, in their hindgut. Whereas if we look at the activated ovaries, it's nearly 84%, which had no pollen at all in the hindgut. So it seems like if we look at how they supply their pollen or the protein resource to actually activate their ovaries, they're rather being served than actually looking for it, which is, I think, the easier way. I mean, delivery might be easier than actually cooking yourself. Nevertheless, it's not always a question if you get the right stuff then. So if we look at protein sources, does it actually matter? So as I showed you a picture of different, of the comb, there's different kind of pollen on the comb as well. Obviously, that corresponds to where they actually foraged and where they actually collected their protein from. And if we look at that meadow, we have a lot of different flowers which have different kind of pollens. For example, if you look at the sunflower, protein content of the pollen there is between 13 and 15%. Canola or canola, for example, has something, depending on the variety, between 11 and 27%. And I put in something Aloe, which is winter flowering in South Africa, especially in Pretoria, has something around 31%. Just as a comparison, here you see a queen cell with a larvae swimming in that jelly which I described. That has a protein content around 37%. Oops, sorry. Does the source actually matter? So there's done stuff, there was research done by Kleinschmidt and Kundus in the 70s already saying that you need a certain percentage of protein actually to do some brood rearing, so even to activate your ovaries then. Herbert said that the optimum level is somewhere between 23 and 30%. And Schmidt said it's not so straightforward. It's not just eating protein. If you eat too much, you actually die. And it depends on how much you actually eat and during which period. So they concluded that protein content actually plays a role as soon in the first 20 days, and the total amount plays a role as well. A study, more recent study, actually found that if you look at the hemolymph and the protein content in the hemolymph and analyze that, the mix of the floral source actually plays a role. But we looked into two specific sources, sunflower pollen and aloe pollen, and looked how that affects the activation of the ovaries. And normally you would expect, since it's 31%, it's close to royal jelly, it should be actually really great to eat on aloe pollen. And if we look in the field, that's actually the case. So the highest development, if we compare sunflower and aloe pollen, is around the aloe pollen, stage 4 up here. However, if we move it into the lab and actually do it under more controlled condition, suddenly that whole picture changes. So it's not anymore that the higher protein content in the pollen plays a role, but other things seems to play a role. In that particular case, our first idea was have a look at the carbohydrate ratio, carbohydrate protein ratio, sugar and proteins, and see if that plays a role, since bees are feeding on quite a number of pollen sources and nectar sources in the field, so that could actually be some kind of explanation behind that. So what we did, we put them again in cages. In this case, we gave them no choice experiments. So we gave them casein as a protein source that is from cows derived. We gave them jelly, and we gave them aloe pollen. On the right-hand side, you see the survival, and you see there's quite a lot of variation depending on the ratio between protein and hydrocarbons as well. That was done by one of my honor students quite a while ago, and she dissected quite a lot of bees as well. But I want to direct your attention to the ovarian activation, and as you can see, sucrose alone has the lowest score, and royal jelly in a 1-to-3 ratio seems to do the best. The other sources seems to be quite intermediate as well. So we find an interaction not only in the ratios, but as well in the sources. To confirm that it's actually the ratio, we took it a step further, and we actually applied Raumheimer and Simpson's geometrical approach to integrated nutrition. And that I have to mention Ruth Archer, Jerry Wright, and Sue Nicholson, with whom I worked on that project or that aspect. And we actually looked at the idea of, are bees actually able to regulate their intake and actually come to a balanced diet? So if you look at the geometrical approach, is more or less the framework behind it is more or less the idea that if I have 2 macronutrients, and you can substitute that even with, you can add that even more, but you have a geometric space. And if you eat a balanced diet, your target is here, what you want to reach in the ratio between carbohydrates and the protein and the amount. If you eat a balanced diet, then it's quite easy to reach because you eat a balanced diet. You get what you want and what you need. If you, however, eat an unbalanced diet, say, for example, one which is more rich in sugar, you would go along that trajectory, the red one, and never get to that target area. So you would be insufficient in protein and might overconsume on sugars. The same the other way around, if you have another unbalanced diet, which is more on protein, and where you miss the target for sugar, but overindulge on proteins. The third option is, if you give them both 2 unbalanced diets, so to say, are they able to actually regulate? So zigzag their way, eating something of the green diet, then of the red, green, to actually reach the target. can they actually know what they're taking in and actually adjusting to what they're getting? And that's one of the studies Salomon did. He's now back in Ethiopia doing his master studies. He looked at different sources of protein. In that case, we used royal jelly again, and the milk powder, since it's easy, or casein, and commercial supplementary, which was advertised as vegan for bees. And we find that independent of the protein source, they actually can adjust their intake. So they're regulating what they actually want to get. However, despite the fact that they get the right ratio, so to say, if we look at the ovarian activation again, royal jelly, that's the part here, the white part of the bar, had the highest proportion of activated ovaries. So it's not only that you eat the right amount, it's not only that you eat the right ratio of a certain of proteins. No, it seems the source itself plays a role as well when it comes to ovarian activation. So great. Nothing really to a certain extent surprising, since the queen is getting the jelly as well, but being fed the good food is the way to go, which is really, I mean, what we would have assumed as well. But we're talking about a social system where one is dominating reproductive activities, and that's the queen. So how do they actually regulate, or how do they actually manage to get these good foodies? And in that case, we have to look at two honeybee pheromones. And you see a drawing of a honeybee worker with the different glands involved. And I focus on 4 important ones. That's the hypopharyngeal glands, which is the food gland, the mandibular gland, Dufour glands, and the turgor glands. The turgor glands, for example, find in African workers, but not in European or workers of European subspecies. And if we look at the mandibular glands, it's one of the substances from the mandibular gland is one of the first ones which actually the function of a pheromone was nicely described and shown how it works. And if we look at the queen, the main components are 9-oxytodecanoic acid, or 9-ODA, which I will use in future, 9-HDA or 9-HDA. So there's 2 aromatic components as well playing a role. And if for the main components for the workers is 10-hydroxy-2-decanoic acid and 10-HDA and 10-HDA. So to remember from that one is that the queen main components are these 3. The main components of the workers are these 2. Nevertheless, they can have in certain ratios as well from the opposite. So the queen can have some of the substances below, and the workers can have some of the queen substances. Why is that the case? So I color-coded it. The purple ones are the queen pheromones. The green ones are the worker pheromones, and that's the aromatic ones, which we find in the glands as well, but which are somewhere in between. With that kind of pheromones, the queen is regulating quite a lot of behaviors and physiological processes. So from retinue behavior, having the court around, to swarm clustering, to mating, it regulates reproduction of the colony, it regulates reproduction of the individual workers, but as well activities as comp building, brood feeding, guarding, and foraging. Sorry. So it's a quite multifaceted or multi-tool, if I want to phrase it like that, when it comes to organizing the colony. Where do they come from? And there it becomes really interesting, because for both workers and queens, it starts with stearic acid. And the only difference is if you go the green pathway, then you produce pheromones which make you smell like a worker, and you're probably a worker. Or you go down the left side and the purple pathway, and then you smell like a queen. You're most likely a queen, or as you will see, a false queen as well. So starting with the same starting product, just depending on one hydroxylation process, if you go the worker side or the queen side. Just on the side, for example, one of the substances I mentioned, the jelly, which the workers are producing, has quite a lot of antibacterial and antifungal activities. Obviously, if they produce jelly, which is rich in protein, would be a nice growth medium as well for bacteria and fungi, for example. So it has antibacterial properties as well. But it as well, and in that case, I refer you to the work by Anja Buttstedt, it's as well controlling the pH and viscosity of the larval food. Just to have a little bit of discourse in worker in colony reproduction, what we see here is a comb with larvae, some eggs, some are sealed, and here we have queen cells. So they're actually facing down. So what we see here on the right-hand side is actually we're looking up onto the ceiling of the cell where that jelly is and where the larvae lies in the jelly. So the combination of the proteins with the fatty acids from the mandibular glands results in a change of the viscosity, which allows the larvae actually to be on top of the jelly. It doesn't drown in it and allows it to actually hang on the ceiling. I made the comparison to one of my students. That would be like you're lying on your pizza and your pizza is hanging under the ceiling and you're eating while you're glued to your pizza. Really strange picture. I don't know how I came up with that one. So how do you get it? Because as I said, the workers can convince other workers and smell like a queen, but she might be a little bit upset about that since she is organizing the court, so to say, and wants to dominate reproduction as well. Coming back to that process, what's happening in laying workers or in workers which start to reproduce would be quite easily. Instead of going down the queen pathway and you produce only a little bit of the purple pathway, for example, the easiest way is to convince other workers to feed me like the queen is actually to switch around. And some of them will do that. The problem then is you smell like a queen. You might attract the queen or the response of the queen as well. That was at that stage only just an idea of what we thought which we can do. So we thought, can we actually make false queen? False queen was described by Sakagami in 1957. And you see here false queen here, and she is actually attended, as you see here, by other workers as well. Can we actually make false queens? So what we did is we took workers, normal workers, and we put some pheromones on top of them. 2 different kinds. Don't want to go into detail. And we put them back into our cages. Here you see one of the workers with the pheromones on the back. And we thought, let's see if we can actually make them into false queens. And if we look at the pheromone production, we used Apis mellifera scutellata, that's the subspecies in the northern parts of South Africa, and Apis mellifera capensis, which is from the Cape of Good Hope, southern parts of Africa, in that particular case, a really aggressive, reproductive aggressive clothing lineage. And what we can show is actually the ones which get the pheromones on the back develop even internally pheromone signals which are similar to a queen. So we can actually quite easily create false queens by making sure that they are perceived as false queens and then get fed, and then they activate their pheromones and even their ovaries as well. So when we see here with extracts, suddenly the proportion of individuals with ovarian activation are actually jumping up. If we look at the results which Mark Schaefer put together, I didn't mention the part which is color-coded by gray, black, and white, and that is how the workers are actually smelling. So black parts of the pie are actually indicating that they're smelling like a queen. And we see the proportion of which have no pollen in their guts, 65% of them actually had activated or developed ovaries. So that strong link in the mandibular gland signal being smelling more like a queen seems to result in the fact that you are treated more like a queen and you get the good food which allows you to actually activate the ovaries and become reproductive active. Nevertheless, I want to emphasize that there is still some workers which have large amount of pollen and still smell like a queen. So there's quite a lot, there's some kind of phenotypic variability as well. However, the problem is a queen does what the queen does. As soon as there's another queen, we see the picture here, thanks to Jochen Flugfelder for taking that picture. If she smells another queen, they actually engage in lethal fights and they kill each other, or they try to kill each other until only one survives. That picture down here is from a YouTube video which I found probably from a beekeeper in the US, where he is really surprised that the virgin queen over here is running around and killing workers. Without knowing anything else about the colony, my suspicion would be that this colony was queenless for too long, so to say, and some of the workers actually started to smell like queens, and as soon as you introduce that virgin queen, she goes around and kills any kind of potential competition. So smelling like a queen, despite the fact that you get the good jelly, might be pretty unhealthy for you because the queen might kill you. How do you counteract that? The easiest way is to actually stay away from the queen, and that was work done by Robin Morris and Robin Crewe and Randall Hepburn, where they looked at the two different subspecies in South Africa again, and their natural hybrid, and they found that more reproductive, more active ones are actually staying further away from the queen. So if we look at that figure here, Scutellata, Apis mellifera scutellata, is relatively less reproductive active compared to the natural hybrid, compared to capensis. So we have that increase in reproductive activity. And what we see at the ratio of, the higher the ratio here on the y-axis is, the more queen-like you smell. You see that they're actually staying away from the queen. The queen was caged, so a score of 1 would mean you're standing on top of the queen. A score of -1 for that particular worker would mean that you're totally on the other side of the comb and actually making sure that you're staying away. And you see that trend to actually stay on the other side of the comb, away from the colony, which is an easy mechanism to not getting killed by the queen. And it as well neatly explains as well why we find brood above the queen excluder. Let me explain that. Normally, bees come, or not normally, in most cases, if it's a commercial setup, bees come in boxes like that. You have brood chamber down here and you have a honey chamber up here. To avoid that the queen is actually going up and laying brood up in that honey chamber, beekeepers put in a grid which allows workers to go through, but keeps the queen at the bottom. Nevertheless, sometimes they're found in these frames that you see here, drone brood among their honey, and they're really upset how the queen managed to get up there. The conclusion would be actually that it's not the queen which went up there, it's workers which are laying up there and staying away from the queen. So smelling like a queen, getting treated like a queen, trying to avoid the actual queen. Instead of laying the brood in the brood area, you're creating your own niche, so to say, and hope for the best up away from the queen. Since I talked already about South African situation or South African honeybee models and why they're so fascinating, let's go into more detail. That's the map of South Africa. Here you have Cape Town, here you have Gauteng with Johannesburg, and Pretoria would be somewhere here. And in the Cape, you have the Cape honeybee, indicated by red. That's this one here. Or you have the Apis mellifera scutellata, which is in the yellow area, actually reaches up to East Africa as well. And in between, you have the interregression zone. The nice thing about capensis is that they can lay a lot of eggs quite quickly. They activate the ovaries and they smell like a queen quite quickly. I mentioned earlier when we looked at the queen, the spermatheca, so the organ actually which can collect the sperm of the queen during the mating flight. And capensis is one of the subspecies where you find the spermatheca in workers as well. So we have a strong we have an organ which is strongly related to reproduction in terms in the way of the spermatheca, still present within the worker phenotype, which I think is at least a good indication that reproduction is not black and white as one might think. What happened in South Africa after 1990? Not only free and fair elections, but beekeepers brought bees from the Cape into the northern parts and then continued to sell it. All these abilities of the Cape honeybee to smell like a queen, to activate the ovaries quite extremely quickly within a few days. For example, capensis takes 9 days, 6 to 9 days. European subspecies, for example, from in Central of Europe or North America, need something between 20 and 30 days to actually get there. And their high potential to actually lay eggs make them really reproductive dominant. That was the word. What happened is then, and that's the result of what we see in the Cape honeybee as well, is that the Cape honeybee is actually able to lay diploid eggs. So if she lays an egg, it undergoes meiosis before it gets split. However, then 2 of the pole bodies actually merging back together. So you get the whole set of chromosomes back. So as we know from Hymenoptera, diploid eggs or eggs with 2 sets of chromosomes developed into female. So you have an identical clone of that particular worker, which makes the clonal lineage more or less reproductive active and more or less lives forever. So that resulted in the fact that the situation in South Africa looks quite different. We not only have 2 interesting model systems, Apis mellifera scutellata and Apis mellifera capensis. Now we have a reproductive active clone as well, which is quite aggressive. And I want to emphasize something. It was around, okay, don't quote me on the exact numbers, but it was several hundred of colonies. And only one worker, so to say, managed to dominate all other lineages in these 250 colonies to actually become that clone. So if we look at the whole of northern parts of South Africa, we have one genetically identical clone doing a devastation among the more commercial colonies. Because the problem is, as soon as you start reproducing, you're not working anymore. So you start only working, you're only reproducing. So these colonies dwindle down and die quite quickly. At the height of it, they were dying within 6 weeks. So there's an economical part to the whole problem as well. So how does the queen keep the workers actually in check, even in these clones? And that's done by work by Fiona. Mainly, we're looking at gene expression levels there as well. We're coming back to that pathway, and what's actually happening is, if we look at it in that aggressive one, it seems like that they're producing some of the precursor to the queen pheromone. So they're getting actually blocked at that stage. So if we look at the gene expression under queen-right condition, that's QR standing for, that is not really expressed. But as soon as you take that social control from the queen away, or from the queen signal, so if the if we have queenless conditions, that jumps up, and 9-HDA actually gets then converted into 9-ODA into the queen substance. Just to correspond what we see on the gene level as well. On the pheromones, under queenless conditions, the pheromones are close to a 1 ratio, which means that they smell definitely like queen. Under queen-right condition, it's regulated, and actually the ratio is sitting quite low. How is it happening normally? Normally, it happens much, much earlier. So if we look at other subspecies like we have in France or in the US or South America, the regulation is actually happening at that level. So instead of going down the purple pathway or the green pathway, it's regulated here. And that's another work done by Fiona. In young bees, nothing has changed. It's not going the green way, it's not going the purple way. But as soon as the bees are a little bit aged and queenless, you see that they're actually jumping and going down the way on the purple pathway. So it seems like capensis is unique in that case, or not capensis. So far, what we find in capensis suggests that reproductive active ones are bypassing that level of regulation, which works in most cases, and have to get regulated at a lower or closer to the queen substance step. Other pheromones, it's not only, since there's a debate if it's a redundant signal or not, other pheromones play a role as well. I mentioned the Dufo glands or the Turger glands. In that case, it's Ola Okosun's work doing a PhD. And we find that it actually corresponds. If from your mandibular glands already smell like a queen, we find similar patterns as well in the Turger glands. As I mentioned, that's actually another trait which we find in both African subspecies we looked at, that these glands are present in Apis mellifera, scutellata, and capensis, but not in Europe, not in most European subspecies. And again, that corresponds to the ovarian activation as well. So smelling like a queen makes you a queen in that case, and allows you actually to reproduce and skip that or challenge that monopoly to a certain degree of the queen. So I'll call it the 50 Shades of Worker Reproduction and Natural Range, and that figure is from Fiona's review as well. If we look at the false queen, that is the one up here, we have mandibular glands, which make you smell like a queen, which is queen-like. The Turger gland secretion is the same, and you can actually lay eggs. You have activated ovaries, and Dufo glands plays in there as well. An incipient false queen has the whole set of pheromones, smells like a queen, but is unable to lie. So we find that quite often as well. Laying worker is the other way around. It smells like a queen. It smells like a worker if we look at the three different sources of pheromones: mandibular gland, Turger, and Dufo. But it has actually activated ovaries. And keep in mind, it would be a good alternative strategy actually if you want to reproduce, because you don't give your queen or any other worker a chance that they can actually smell you to be reproductive active. It might result that you might have to get your proteins from another source than from the jelly, but it might increase your survival actually to be able to lay eggs. And then you have subordinate workers, which we would classify as a classical worker, smells like a worker, has inactivated ovaries, and probably behaves like a good worker and is not reproducing at all. Is that only something which we find in capensis and scutellata? As I said, capensis scutellata is stretching up to East Africa as well. So here we have the natural distribution area of Apis, so from Cape of Good Hope to the northern to Norway. If we look at the pheromones, and in that case I have to say African honeybee subspecies are actually better researched than European on that one. We find in most of the subspecies we looked at some kind of 9-ODA in their mandibular gland pheromones. In some only traces, but that is all under queen-like conditions. So you would think the queen has everything under control. It's making sure that the workers are not producing a signal which is similar to the queen, or which let other workers perceive that particular worker as queen, as royal, so to say. But we still find traces there. So I would be I would say that that is quite a good indication that we have probably more level of reproduction in these kind of subspecies as well. So if we look at the literature, especially at policing as well, do we have actually a gradient of worker reproduction in the natural endemic range of Apis mellifera? So is there no worker reproduction up here, explaining all the results on European subspecies, that worker policing is efficient and that there's only a low number of eggs, if at all, surviving from workers? Or is there any other potential reason behind it? The red areas are the invasive range, just to explain the different colors. But let's focus on the native range. I think the big difference, and sorry, come back to that one, is if we look at the northern parts, and that's the striped area, it's predominantly managed population. So if we pick a colony or if you pick a bee in Germany or in France or in Spain, we probably can assign a beekeeper to it. So the number of colonies corresponds with the number of beekeepers which we have around. 90% of the bees in Africa are actually wild. So if we look at the colony cycle, what is normally happening? The queen is laying eggs, the population grows. If the population in the colony is big enough, you have a split of the colony. So the old queen leaves with the proportion of the colony and tries to find a new nest. Virgin queens are raised in the old place, so to say. When they hatch, the queen fight. The virgin queen, which survives, goes on a mating flight, and then the circle starts again. And normally people are focusing on that part and ignore the swarm here. So what's happening in that swarm? Obviously, that has a lower percentage of survival since it has to find a new place anyway. And during that travel, which is several cases, there's a probability that the queen might die as well. As well, it's the old queen which is leaving, so it might be just age. So as soon as the queen dies, you have a change in the relationship among these workers. I mean, suddenly it becomes, from a relatedness point of view, not anymore beneficial to help to raise nephews. It's rather beneficial to reproduce yourself. If we then keep in mind that the mating frequency is in African honeybees is above 25, and capensis up to 54, you have a patchwork family there, indicated by the different colors here. So you have actually quite a strong pressure to reproduce. So what I'm suggesting is that the fact that you have swarms which are more often issued and not controlled for, you have the pressure on workers to actually be ready for the situation when the queen disappears, to actually then reproduce and ensure that you get some kind of genes into the next generation. So I hope I made the case that worker reproduction is part of a colony cycle in general. Especially African honeybee worker seems to produce queen signal, even if the queen is present, so that somehow avoid her and challenge her monopoly. We can induce the transition to false queen quite easily with an external pheromone. Other sources play a role as well. So there's a whole potpourri of different glands playing a role when it comes to investigating the role of reproduction, I think, in honeybees. There's a lot of people to thank. Robin Crewe and Robin Moritz as one of my earliest and longest mentors as well. All the different students involved, Hanneli Human, for example, but I mean, Ruth Archers, Sue Nicholson and Jerry Wright from Oxford as well. My late supervisor, Prof. Randall Hepburn, and I'm really enjoy that picture all the time of his. I'm happy that I don't have to do beekeeping without a protection in African bees because they're quite defensive. And I'm even more happy that I don't have to wear a tie to do beekeeping. Yeah, there's a lot of people worked on it, and there wouldn't be the results without them. The same with all the funding. And yeah, thanks a lot for your attention and for listening.
Yes, thank you very much. A great talk. I mean, it's so fascinating to see this entire new world of the honeybees again, and but with very similar questions like we're asking in all these other fields of social evolution. Very fascinating. To remind everyone again how we do the question and answers. If you want to ask a question, then please type a question mark into the chat function, so we see who wants to ask questions, and then I will call up on you. You can have an idea when will be your turn. And even if you have been asking questions before you ask your question, please first shortly introduce yourself, say what is your name and where you're coming from, so we know who we are talking with. There are no questions yet. Also for the people on YouTube, if you have questions, you can write it in the chat and we will transfer it here. So I can ask with, maybe I was a bit also distracted as I was looking on YouTube, or maybe it's because I have a mammalian brain and don't understand social insects. Can you explain me once more, why do we have some so-called false queens and egg-laying workers? What is the difference between the two, and when does this difference occur? Is it something early in development, what food they get as larvae, or why are there these two types of females?
I'm not sure if it's the food during the larva stage. I mean, there's some work from Poland suggesting that the queen status during their development plays a role if you become more reproductive active or not. But in that particular case, I think it's just a variety of worker phenotypes which we can find in the colony. So some of them just have the set to actually become false queens, to have the whole set of smelling like a queen and actually reproducing, and others just don't have the pheromone set but are able to activate the ovaries. So if that is food related, not sure. We know that it's mainly young bees which are able to actually become or that age plays a role if you actually become a laying worker or a false queen, and that it's, if you reach a certain age, then it's over. But the mechanism behind it, it's still battling to understand it. But you could find false queens and laying workers and the queen in the same colony.
But the phenotype of the false queen, is it just chemical, and then you see it from the behavior, or can you see morphological? Oh, that's a false queen, and that's a worker.
No, you would have to sample the pheromones, or you have to be invasive and actually cut them open and see the ovaries. You couldn't say because it has more or less hairs on their pollen basket, for example, if that's a queen or not. For example, we used silicone, or that's a method Robin Crewe developed. We used silicone tubes actually to rub their mandibles like a toothbrush. We call it toothbrushing, where we can actually take up the pheromones and then see what they're producing. That is a non-invasive way where we could look at and see what's happening.
Good. Then I have one more question before we come to Gloriana, because I really would like to know this. You have these two subspecies of honeybees in Southern Africa, and one sees the natural distribution range. Like you said, most of these are wild living there. What keeps them apart naturally? I mean, I've seen, of course, I know South Africa has a work there. The Cape honeybee is maybe in the Cape Floral Kingdom, which is relatively cold winters, and it is a winter rainfall area in contrast to the rest. But in Namakwa land, it's also winter rainfall that is botanically similar. You have the other bee species. Do you have an idea what keeps these two subspecies apart from each other?
I think that's a really good question, and I think that's one which is intriguing all of us for a while already. My guess would be actually it's a the kind of being, how aggressive you are in terms of reproduction. Because if you look at capensis, for example, and it's quite difficult to pick up, since if a worker lays a diploid egg, it's a copy of itself. So to identify if that egg was laid by a worker or by a queen is rather tricky. So you would actually have to observe these worker reproduction. But nevertheless, you can pick it up if foreign workers are reproducing. And it seems like that you have that problem that workers are reproducing and trying to take over the colony. What happened with the clone in the northern parts of South Africa? That is happening actually as well within the capensis endemic range. So they're trying to take each other over, and as soon as they hit a capensis queen, they get regulated and nothing is happening. If they would hit a scutellata queen, they would take it over and destroy it. And that's the way it's actually stable, that region, I think, because as soon as you have capensis trying to move further north, they're hitting a suitable host, but the density of the hosts are too spread apart. So instead of getting the next host, they're actually dying off. And in that way, they can't really spread forward, further up. And Scutellata can't really spread further down, because as soon as you would spread further down, you get nailed, if I want to phrase it like that, or taken over by these capensis, aggressive reproductive workers, and you die out. So in that way, you have that, I can't go further north because I don't have enough hosts. The other one can't go further south because I'm then getting taken over. For example, we have histories of introduction of European subspecies into Africa because people, especially NGOs, thought European honeybees are better or better suitable for that. There is no single anymore of these left. So if you bring in European honeybees, they get taken over by capensis and they're gone within a few years.
Okay, that could explain why the capensis doesn't succeed in the Makwaland, even though it has also winter rain and a more similar flora, but it has much less flowering plants, and thus probably densities are much, much lower than the Cape Flora Kingdom.
I think that's the explanation, yeah.
Yeah, that makes sense. Then we have a question by Gloriana.
Hi, I don't know,
Hi.
I hope you can hear me okay. Thank you for the talk, Christian. Very interesting. I also come from a mammalian background, so I hope I understood everything correctly. But I had a question because at the beginning you were saying that in order for the bees to develop the ovaries properly, to be able to reproduce, that they had to be fed this more adequate diet, right, from the others in the colony. So then my question is, how does the laying worker develop this, more proper ovaries if they're not producing the pheromones that are making the other workers feed them?
My educated guess on that one is that they're actually feeding on that's what I wanted to show with the picture where you, on the frame where the pollen actually is. I mean, you have a protein source which is readily available for the workers. So they could just eat on that pollen like you used it as a supplementary as well and get the protein resources from that. The only disadvantage is they would have to digest it. So their hindgut might fill up, which is not really a problem because they could fly out and actually get rid of it. So it might be just that they're not as efficient in becoming or producing as many eggs as one which is fed on royal jelly. But that's a speculation from my side. So we didn't really quantify if you only give them pollen, they're only producing 10 eggs, for example, or if you give them royal jelly, they're producing 20 eggs. So that's the explanation why you find laying workers and how they actually supply themselves with the protein source.
Okay, thank you.
Pleasure.
The next question comes by Maren Hook.
Thanks for your talk. My name is Maren. I'm from the University of Derby. Again, I'm also from the mammal side, so I might have missed some of your explanations. So I have a double question. So firstly, do workers also mate with drones? So do they also produce fertilized eggs? Never.
No, we tried that. We tried to do artificial insemination on the Cape honeybees since they have quite a lot of, or the workers of the Cape honeybees, since most of them have a spermatheca, but we didn't succeed. And yeah, there's no. The honeybees are mating in mid-air, so they would have to fly to an area where drones are, and the drones actually would have to recognize the worker to be something you want to mate with, and then the worker would have to carry the drone since the female is doing all the work during mating in mid-air. So from the behavior, impossible. And it seems like from the even from the physiology, trying to artificially inseminate, it's not possible. Okay, that was a long answer for no, but yeah.
No, that's great. So that means if workers lay eggs, they lay sons, and that obviously shifts the sex ratio. And as far as I remember, there is some discussion on who's winning, whether it's the queen winning or the workers winning in terms of kin selection and so on. So if the sex ratio is changing, how does it change the whole interactions between workers and queens in terms of who's winning, how they try to manipulate sex ratios? Is there any study on the behavior that is following from that, depending on how many workers have been laying?
Not really, since the queen is still outcompeting a number of eggs laying. So I think the effect on the sex ratio itself is rather minute, if I want to phrase it as well.
Okay.
Also, drones are only produced, or mostly only produced during a certain period of time in a year, during the mating season normally. So I would assume that the queen is then laying drones as well. So again, the sex ratio isn't really so much affected. But honestly, I haven't really thought along that line, and I will now think a little bit more about it. But as I said, workers are not participating in hive duties as soon as they reproduce, and the queen would kill anything which remotely smells like competition.
Thanks a lot.
Pleasure.
So the next question comes by Suleima.
Yeah, hi. Suleima Tang Martinez, University of Missouri, St. Louis in the United States. And I find the examples you gave with the capensis, with the clones, really intriguing. And I have a bunch of questions because I'm not sure I'm really understanding exactly how it works. And so I'm going to go through all the questions I have rather than asking them one by one. So
yes.
I wonder whether these false females, the ones that start the clones, do they start off in a normal hive and then in a normal colony and then go off and found a new clonal colony? Do they produce females the way you explained through meiosis and then the two eggs fusing, but do they ever produce haploid males, or are they always all female clones? Do they forage, or do you have any sort of reproductive division or division of labor in terms of anything that looks like a worker caste versus the queen? And is there any evidence related to that the queen can inhibit the other daughters or clones of her, or try to inhibit them? I don't know if that's all clear.
Yes, thanks a lot. It's all clear. To switch on my microphone. Okay, answering the easy question first. They're not participating in hive duties, which is the problem as soon as you have a whole bunch of them reproducing, the colony collapse. Are they reproducing in their own colony? As I said, that's difficult to find out genetically, since we don't really know who laid the eggs. But it suggests that we know that they're reproducing in other colonies of capensis. So they're actually going out and trying to reproduce in other colonies. There we have genetic evidence that a foreign worker reproduced, or foreign workers reproducing in colonies. That was reproducing, that was a foraging. Sorry, what was?
Whether they ever produce males anytime, or is it always by definition, if they're clones, they're producing females, but I wonder if they're also capable of producing essentially unfertilized eggs that would become males. And then do they do their own foraging?
No, they don't.
And is there any evidence that there's any effort by, let's say, the founder, if we want to call her that, of trying to inhibit the others from reproducing?
Okay, there's a strong signal from the queen to actually prevent that kind of production. So queens of the Cape honeybee are quite able to control them, or to a better degree than any other queens. So if you compare that with European subspecies or even any other queens, they lose out there. So the queen is able to actually control them to a certain degree, not fully.
Even, Christian, even in the clones?
Even in the clones.
Okay.
So, but not as good. The clones have another disadvantage, which I mentioned just now. If a worker lays diploid eggs, she will always lay diploid eggs. So that's not really switching there anymore. But you could have a colony where you have some workers in the Cape, for example, where some workers lay diploid and some workers lay male. So that depends on the individual which you're picking up. What we see in the clone is extreme, since it was facilitated by humans as well, is a strong selection for actually being aggressive in terms of reproduction. So you have actually short-sighted selection, like with your flu virus. They trade transmission for virulence. So they're highly virulent, but their transmission is really bad. So if we look at the clonal lineage, they're really, I think if the beekeepers would listen more to the researchers, we wouldn't have that model system anymore. That would be solved long ago. But now we are approaching 25-year anniversary. So thanks to everyone who's helping to maintaining it.
Sulema, your microphone is off.
Do they in the clonal colonies, are the individuals in it foraging?
No. What you see is normally you have a host colony which is taken over, and then these clones are just eating.
Thank you.
Pleasure.
Fascinating.
Yeah.
Thank you. There are other questions coming. I just want to add here one question as it fits for my understanding. You said the capensis that was brought by beekeepers from the Cape to other parts of Southern Africa, in the meantime, they are all clones. So that means from the eggs that the clones lays, there are normal workers hatching. Is that correct? And then how do these clone colonies reproduce?
The clone colonies need a host. So it's like our flu virus. If they don't have a human host, or stick with the human host. If they don't have a human host, they're not able to reproduce, full stop. And that's the same with the clone. If they don't get a scutellata colony as a host, if they don't find that one, then they're gone. That's why it would be an easy solution if you make sure that the transmission is stopped.
Okay, now I understand. Then the next question comes by Quentin.
Yeah, hi everybody. I'm Quentin. I'm come from University of Pretoria. So I'm glad to see Christian talk, because I usually see students, so I'm not gonna miss the opportunity to ask a question. Christian, I wanted to know, I'm always amazed during this talk to see how much a pheromone can change the physiology and the metabolism of the bee. And I was wondering if a false queen or laying worker can turn back into a normal worker. And in the same line of thought, did you ever try to make false worker by putting worker pheromone on a queen? I know queen are rare and you don't want to waste them, but I was wondering if it's possible.
No, then okay. First question. No, they're not reverting, so they're going that way and then they stay that. I think you would lose against the queen. What we see, what Vincent Dietermann did in his work is actually if you compare two workers, they're actually competing for pheromonal dominance. So the one is increasing, the other one jumps back, and only after 3 or 4 days you have actually a winner declared, so to say. So if you put a worker smell on a queen, I think the queen will just up their pheromone production to smell like a queen. It will get challenged. It will feel challenged that somebody is there, or I'm not really getting the right attention. Worst case, the workers think it's a worker and kill it, but I have my doubts. We're talking 10-fold higher, more pheromone production, 10, 20-fold.
Thank you, Christian.
Pleasure.
Next question comes by Eduardo.
Hi, thank you, Christian, for a fascinating talk. And I'm gonna, it's not a question, I just would like to ask if you can please share with us a little bit of this fascinating intersection that you are facing between pure science and behavioral ecology and research on the behavior of bees, and this very, very applied commercial activities around the beehives and all that. So some of the situations that South Africa is facing that you described to us really are at the center of things that we've been discussing for decades: the importance of animal behavior, the applied importance of doing basic behavioral research and basic natural histories. We always talk about that, but it is not always the case that you have compelling examples of how the kind of research we do may be of relevance to economic development in a country or economic activity. So I was just curious if you feel like sharing some more of what's going on and the extent to which you as a behavioral ecologist is contributing to that.
I have to say, especially dealing outside science with beekeepers or so, it's always an uphill battle, because you have to actually try to convince them to believe into the science. And I think it's similar to the pandemic as well, where we fight with different players involved. I mean, one of the solutions to solve the capensis problem was to stop pollination services, so to stop bees moving around in the country and thereby reducing transmission. Sounds familiar, I guess, and making sure that the clone is actually disappearing. The argument from the industry was we have to pollinate; it's our financial income, and we have to actually continue with it. So the government wasn't listening to the researchers or scientists, and rather to the industry. So we have it still running. So it feels like, actually, if I make that comparison, and I actually will credit you for giving me that idea. I think we are in the 6th, 7th wave now of the pandemic, which is called a clone, and nobody's actually following the normal rules of reducing the transmission. So I think understanding behavior ecology of your target animal more and taking it into account when you have a business operation or even just conservation issues, I think is crucial, because otherwise things like this happen. So I think there it comes that we actually, as behavior ecologists as well, make sure that we communicate our results to the greater public as well, in a more understandable way. So without that basic research, we wouldn't be even closer to a solution.
Good. I have if there was I think that was the answer to Eduardo's. Or Eduardo, do you have something else to ask? Otherwise,
No, I just was curious to see Christian's reflections on the situation and whether there was anything, but unfortunately, I guess it is a very, very rosy landscape of the dialogue between researchers, and which of course doesn't surprise us, but I was just curious. Yeah, no, thanks.
I mean, I think that's a great comparison to, if one sees these and parallels, people not listening to scientists, doesn't matter if they are politicians, people that want to ignore the pandemic, or if they're ignoring the dramatic effects this can have on natural pollinators, because these honeybees, they are naturally. I had one other question, and this then comes to one more question of my overall understanding, because I have the big big gap. You repeatedly said that the queen is going to fight with reproducing workers. And from what you said, it sounds like the queen is kind of always winning. Is that true? And is this because she a question of size, or did I get this wrong? And does the worker have a chance to win?
I think that is a good question, because we don't really know how the host queen of Scutlata, for example, is lost. I think the point is the chance of a worker winning a fight is not zero. And since it's not zero, at one stage, one worker might win. And that is, I think, the way the clone workers actually overcome the host queen. It's not, if you look at just one worker, that one doesn't have a chance, because the queen is bigger, is stronger, and is actually tuned to actually fight and kill. So if we look at 1-to-1, the worker doesn't have a chance. But if we play the number game, at one stage they will probably overcome the queen. And you have to keep in mind, you have a lot of feedback mechanisms in the colony. So for example, if the queen is constantly running around and trying to fight, she's not laying eggs, which perhaps other workers perceive as that she's not fertile enough. So suddenly they might join and want to get rid of the queen, because they think she is not fertile enough, let's get a new one. So that whole social dynamics can change quite quickly if you have a lot of interactions going on there. Sorry, my son is waking up. Oh, quite late.
That's many of us are used to this. It's a lively world. Now I come to my most important question, because I see I have a deep knowledge missing. That's about the evolutionary or fitness consequences of being one of these clones. You said when the clones take over and they only produce other clones that are not working, and at some stage the colony collapses. So what is the evolutionary fitness? It seems like they're never producing really individuals that will produce new colonies, and with this have long-term fitness. I think I must be missing something here, because otherwise the clones would go extinct at some time if the other colonies always collapse.
Yes, that would be expected. So evolutionary is actually a dead end. As long as they have hosts, they will survive, but as soon as that host is gone, they would be dead. Yes, so what happens is, as soon as there's enough, if the host colony is more or less dead, and there's not enough workers anymore taking care of the clones, so to say, you will see that they start trying to fly around and find new hosts.
Okay, so it's really a parasitic strategy.
Yes.
Okay, and my last question, because it's really my last one, and I know I have none left. And it's an obvious question. The beekeepers prefer capensis because they produce more honey, or what is the reason that they prefer this subspecies?
No, there's actually no preference. The reason why they brought it up to the northern parts was because he was doing pollination in that area, and he didn't want to take it back to the Cape. So he was selling them there and then, and that's the way it spread. It's more or less going in that direction of Eduardo's comment earlier, or question. They were not thinking about the fact what it means to actually move an animal from a native area into a new invasive area.
So there's actually no good reason that they use this subspecies and not the native one, except
no, I mean
historical.
Yeah, because you have big beekeeping organizations which do pollination of apple, for example, in the Western Cape, and then they go to the sunflower fields near Pretoria, and instead of raising colonies in Pretoria, so to say, they bring at that stage they brought the colonies from the apple pollination up to the sunflower pollination.
Okay.
And what we know from all subspecies, especially from work in Europe, is that if I use the South African phrase, local is lekker, which more means that local is local adaptations are there, it's much better to use a local bee than even shifting around from 300 kilometers away.
Okay, I mean, this is really frightening, because what people who don't know South Africa might not know, you have the Cape Floral Kingdom, and then you have mainly the Karoo, a very dry area that can be something like a safety belt with low productivity, low densities of the other bee subspecies. But then in Gauteng, you might have much higher population densities where these clones can really do big damage and will not disappear easily, like it would be in the Karoo, where they can't invade. Because that's quite interesting. Then another question by Suleima.
Yeah, Karsten actually just mentioned part of what I was going to ask, but essentially it seems that what you have is the equivalent of intra-specific parasitism. And I'm wondering if you see any parallels with the situation you get in some ants, where you have interspecific parasitism that, you know, might work differently, but it's still parasitism. And in that case, one species of ant essentially either raids or takes over a colony of a different species. In this case, it's parasitism, but it's occurring within the same species. And also, with regards to what you just said about once the host colony dies, then that particular clone dies with it. If a host colony is dying, can't they fly off and invade a different colony?
Okay, starting from the back again. Yes, I think that's the situation at the end of that whole infestation, or when with the whole takeover, you will then find workers which starts flying around because they're not getting fed or whatever the trigger is. At the height of the capensis problem, so to say, you had really mini swarms. So that was one worker with 20, 40 bees, because most likely the worker smelled like a queen. So some of the workers decided to leave with her to a new colony, and they went to new colonies or try to get into new colonies as well. So they will try to find something, and if the density is high enough, they will go into it. I think I would rather compare it to a virus, actually, because it's not like that these clones have any viable colony. It's not like a slave raiding ant, for example, which takes the slaves and then makes sure that the colony is running there. So intraspecific social parasitism, yes, but I think slightly different from the interspecific which we find in ants, especially for that reason.
Thanks.
Yeah. Yes, thanks again very much for really a fascinating talk. It opened an entire new universe to me, but I also have to say, I see Standive is still in the audience. Standive, if I don't find time to comment on our discussion tomorrow, it's because all night I've been thinking about these two different subspecies of bees, because it's just really an amazing system. But with this, I'm stopping the live transfer here on YouTube, but everybody is still welcome to stay here. If there are still some other questions coming up, either for Christian or questions in general. I see there's still one question coming from Eduardo. I don't know if it's for Christian or a general question, Eduardo.
Yeah, it's for Christian. I wanted you to repeat some of what you said with regards to how you have used the geometric nutrition framework to understand activation of the ovaries. And the reason I want to understand it well is because there is a number of colleagues who have over the years looked at the activation of the ovaries in human females and all kinds of non-human primate females. Now the proxy there has been, they haven't really assessed, like you can do with the bees, the ratio between proteins and glucose, for example. It's more about, do we see a change in the way, do we see actually a change in the trend of where the female is gaining weight or not. And so apparently what they find, and this is very close to the research that my wife has done, Claudia Valeja, so that's why I'm very interested. But also, we were talking about the geometric framework for looking at the nutritional ecology of owl monkeys and trying to understand how changes in the nutrients that we give them may have different effects in their reproductive success. Of course, we cannot. Well, I'm just trying to understand what is it exactly that you did and what you found. My understanding is that you were looking at the ratio between protein and sugar, and that signal of the ratio is what is telling the bees, yes, let's go reproduce or not. Did I understand it well?
I think yes, but as well that it's not the only trigger. So the ratio is important. So if you feed them only protein, they actually die. So they're not really able to reproduce whatsoever. So they don't get to that stage. So they need a certain ratio of proteins to sugar. But then the source plays a role as well, which suggests that perhaps only looking at 2 macronutrients is not enough. The next step would be to include lipids, for example, to see how that plays a role. That's where we took a shortcut, so to say, and used pollen, because that's a potpourri of different things. And we know, for example, from field observation that aloe pollen, for example, is extremely good in actually allowing workers to reproduce. It's quite common as soon as the bees are feeding on aloe pollen in the field. So that was an idea to look if we can bypass that and going into the 3rd, 4th, 5th, or 6th dimension, if we can actually solve that part. But the trigger would be you get the right food, either as pollen or jelly. In that case, probably a ratio of 1 to 12. If we talk about the 2 macronutrients, probably can trigger that kind of activation, yes. But we would have to do some controlled experiments and knockout things actually to prove it, I guess.
Thank you.
Then there is one question from YouTube. Sam Diel is a master student in biology from Humboldt State in the United States, studying foraging behavior in honeybees. And Sam asks, here we are seeing a rise in laying workers in queenless hives. And from personal experience, replacing the queen is nearly impossible. Is there a way to remove or find a laying worker?
Okay, the easiest way is probably, but that might be the most costly way as well. You put virgin queens in there and you let them try to kill the workers off. Obviously, you have to do that constantly and make sure, because it seems from what the question is that some of the laying workers are actually killing the queen. So we're coming to that, the numbers games where you actually don't get in. That would be an idea. Otherwise, queen pheromone, queen pheromone, queen pheromone. So you can't buy queen pheromone anymore commercially, but having an external source to actually get the colony back on track would be an option. But at the moment, I would say put in queens, 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 or merge them.
Yeah, that sounds very expensive.
Yeah, yeah, that's why I said. But yeah, because you can't really smell themselves which one is a laying worker, who's not. But please drop me an email. We can chat via emails a bit more about that.
Okay, great. Thank you very much, Christian, again for a fascinating talk. Thanks a lot to the audience for a very lively, interesting discussion. I think we all learned a lot today. For me, it was really an eye-opener for how fascinating nature and science and animals just are. It's really amazing. Then, like I said, I'm now going, as there are no other questions, stop the live streaming, but you're still